Laboratory-associated infections continue to be serious hazards for people engaged in research or clinical diagnostic work. The magnitude of the problem is apparent from data accumulated during the last 50 years. Since 1925, some 4,000 reported infections have endangered the health of laboratory workers. 160 of these infections resulted in death for the individuals involved. The majority of the laboratory infections occurred at institutions engaged in research or clinical diagnostic work. The people involved were primarily trained professionals, people most knowledgeable about laboratory hazards. The meaning is clear. Infectious hazards can only be reduced by carefully following appropriate procedures. This presentation has been prepared to provide the knowledge you need to reduce biological hazards in the laboratory. Of 4,000 cases studied during the past 50 years, only 20% have been identified as resulting from known causes. 80% of the cases investigated resulted from unknown causes. Most investigators concluded that infections from these unknown causes were due to routine microbiological procedures, including procedures which resulted in producing aerosols which contain microbes. These aerosols were found to be the principal agents contaminating the laboratory environment. Studies conducted since 1950 suggest that the following questions must be considered. What forms do potentially infectious aerosols take? What factors determine whether a laboratory worker becomes infected? What determines the relationship between these factors? We also need to know what common microbiological procedures are likely to generate infectious aerosols. We may also ask what procedures are likely to be hazardous in work with experimental laboratory animals. We have posed these questions to add emphasis to key points to be explored in this presentation. In identifying aerosols produced by routine procedures as principal contaminants of the laboratory environment, two distinct forms have been described. One kind of aerosol is primarily composed of large droplets, larger than 5 microns. The second kind of aerosol consists mainly of droplet nuclei. These are tiny, solid particles whose usual size is less than 5 microns. The two forms of aerosols contaminate the laboratory environment in contrasting ways. First, consider the aerosol composed primarily of large droplets. These large droplets settle in a relatively short time and contaminate work surfaces or skin. The droplet nuclei found in the second type of aerosol form as a result of almost instantaneous dehydration. In contrast to large droplets, droplet nuclei may remain indefinitely suspended in the air, drifting about with even slight air currents. These droplet nuclei may contain only one or a few microorganisms. 4 factors determine whether you, the laboratory worker, will become infected by contact with large or small droplet aerosols. Factors determining infection are extent of contamination, route of infection, virulence of the organism, and host susceptibility. Interaction between these 4 factors will vary with each pathogen handled. This is why the microbiologist must have a detailed working knowledge of any disease-producing agent he or she is to study. The first factor on our list is extent of contamination. That is, how many microorganisms contaminate the air or surface with which the microbiologist comes in contact. These may be measured with air samplers or liquid impingers. The second factor is route of infection. The route may be by inhalation, ingestion, penetration of the skin, or accidental inoculation. The inhalation or airborne route occurs in diseases like tuberculosis. Such diseases are spread by inhalation of bacilli contained within the nuclei. Each droplet nucleus is less than 5 microns in size. Tuberculosis and other diseases transmitted primarily by droplet nuclei are less likely to be of concern when only surface contamination occurs. Ingestion is another frequent route of infection. Salmonellosis, shigellosis, or other enteric diseases are commonly spread in this way. Touching contaminated surfaces can result in finger-to-mouth transmission of these infectious agents. Penetration of unbroken skin is also an important route of infection. Tularemia is an example of an infection that may result from penetration of unbroken skin as well as from accidental inoculation or inhalation. Still another route of infection is through accidental inoculation, which may occur as a result of animal bites, needle inoculations, or punctures by broken, contaminated glassware. We have explored 2 of the 4 factors determining infection: extent of contamination and route of infection. The other 2 factors are virulence of the organism and host susceptibility. When considering virulence, remember that some infections may be initiated with 1 or just a few organisms. Others may require large inocula to initiate infection. Finally, host susceptibility will determine the severity of the illness resulting from infection. Increased resistance to infection can be achieved through vaccination. We must assure that personnel are properly immunized. We have now identified 4 infection-determining factors: extent of contamination, route of infection, virulence of the organism, and host susceptibility. The importance of these factors varies from one pathogen to another. We shall now look more closely at the role of aerosols as a cause of laboratory infections. Potentially infectious aerosols are formed when common laboratory tools are used. Care must be taken with procedures involving the inoculating loop, pipette, syringe, and needle. Frequently, aerosols are produced when the inoculating loop is used to streak an agar plate having a rough surface. Such roughness occurs where bubbles remained in the agar surface while it hardened. Aerosol formation may be significantly reduced by using only smooth, bubble-free agar media. The aerosols may be further reduced by spreading the inoculum with a sterile bent glass rod. Using the bent glass rod reduces aerosols as much as 65%, even on rough agar. The inoculating loop is often sterilized in the flame of a Bunsen burner. When the hot loop is immediately touched to colonies on solid media, or when the hot loop is inserted into a liquid microbial culture, the sizzling creates a dangerous aerosol. aerosol formation may be reduced as much as 99% by allowing the loop to cool before using it. Infectious aerosols also result from the improper use of pipettes. When the last drop is forcibly expelled from a pipette, the bursting bubble generates a dangerous aerosol. To minimize this problem, drain contents from the pipette by gravity flow. This reduces aerosol production by as much as 2/3. The pipette is frequently used to mix culture suspensions in a tube. This often results in aerosol-generating bubbles forming at the liquid surface. You may reduce bubble production by using one of several available test tube mixers. Here, mixing is centrifugal, forcing organisms toward the walls of the container rather than producing bubbles. When the tube is kept closed during mixing, the nearly saturated atmosphere in the tube greatly minimizes droplet nuclei formation. Some large droplets, however, may escape as the cap is removed. Every microbiologist has, at some time, seen a drop of culture suspension fall from a pipette tip. The amount of potentially infectious aerosol thus formed varies with the type of work surface. The harder the surface, the greater the splatter. You can reduce such aerosols by as much as 90% by placing a towel soaked in disinfectant on the work surface. Procedures involving the syringe and needle are also potential sources of aerosols. Do not use a syringe as a substitute for a pipette. Accidentally dislodging a needle from a syringe can be especially dangerous. To prevent this kind of accident, use only locking syringes. When a needle is removed from a rubber-stoppered vial, the needle will vibrate, producing an infectious aerosol. Wrapping the stopper and needle with alcohol-soaked gauze considerably reduces these aerosols. Expelling air from a partially filled syringe also creates an aerosol. This procedure should be done only when the needle is covered with alcohol-soaked gauze. Some laboratory procedures require handling and injection of experimental animals. Laboratory animals should always be handled by trained personnel. Animals must be properly restrained during inoculation. When animals are inoculated, a portion of the inoculum often leaks back onto the animal's skin. When the animal is returned to its cage, organisms on the skin contaminate bedding. These organisms may then be carried throughout the room on air currents. Swabbing the injection site with antiseptic both before and after injection will reduce airborne organisms by 30%. During this presentation, we have explored ways to reduce infectious aerosols in the laboratory. We have seen how large droplets and droplet nuclei aerosols are formed in common laboratory procedures and may result in contamination. Proper use of the inoculating loop, pipette, needle, and syringe will significantly reduce this contamination. Many infectious incidents stem from the use of techniques which create aerosols. By reducing aerosols as depicted in this presentation, you can eliminate many of the infections that commonly occur in the laboratory.